Alright, hello everyone. Good evening to all of you. My name is One Who Gets Bread, and tonight it's actually the second day of college. I started at a new university, so I've been quite busy over the past few days, and I thought tonight I just have a little bit of homework to do, so I just thought, well, why not stream with some people in my community and get my homework done at the same time, so we're kind of doing two things at once here. And so, yeah, I just wanted to talk with all of you guys while I'm trying to complete this. I'm doing something for one of my genetics classes, so it's gonna be some interesting stuff. There's like a YouTube video too that I'm supposed to be watching, and I'll be doing that all on stream, so be right back. I need to adjust the light. There we go. I think that should do it better. Okay, so once again, it's been day 2 of college, and I haven't been streaming in a long time because, of course, I want to focus on my schoolwork, but I'm glad to be back, and hopefully tonight we can get some stuff done while we're having fun streaming. So let's get started. I'm just gonna start watching this, and if anyone else is watching, just feel free to ask questions and chat in the chat room. I'd love to hear from you guys. I haven't been on Twitch for a long time, so it's good to be back. Alright, let's get started.
Hello, and welcome to the help session on complementation. Today, we will briefly be reviewing the concept of complementation, and then we will discuss complementation with a non-biological example. So, to begin, complementation is generally used to determine if two mutations fall in the same gene or if they fall in different genes. So here we have two mutants, mutant 1 and mutant 2. Both of these have mutations in the lysine biogenesis pathway. So on their own, they cannot produce lysine. To determine if these mutations fall in the same gene or in different genes, we need to cross the two mutants together and examine their progeny. If the progeny can grow, this means that the two mutations were in different genes, and thus they complement each other. However, if the progeny cannot grow, this means that the two mutations were in the same gene and they do not complement each other. So now, let me explain complementation yet another way. So over here we have the knitting pathway. It's very simple and straightforward. So in order to knit, you need two things. You need both knitting needles and you need yarn. When you have both of these things together, you can knit a nice scarf. So to explain how this is related to complementation, I've brought along my friend Robert to give us a hand. Alright. So in the first scenario, we're going to have me with yarn and Robert with needles. When I have yarn, I can't knit by myself because I am lacking needles. This is equivalent to having a mutation in what we'll call the gene for needles. Robert, on the other hand, has...
So right now the assignment's just about crossing these two mutants, and so you have genetic complementation, where you're trying to find out where in the pathway did the gene not code for something that's needed in the pathway of this synthesis. So right now they're just using the example of knitting with only needles versus knitting with only yarn. You need both of those things to start knitting a scarf. So once again, like with the genes, if one of these mutant species of the phenotypes that we're looking at, they don't have let's say, gene A that codes for the first enzyme to produce the first product in the chain of reactions. Well then, if you cross that first mutant with another one that has the inability to make the synthesized compound, but it might not have enzyme 2, or it might not have the gene to code for enzyme 2. So then, you can't make that end goal, you can't make that final synthesized compound that you're actually shooting for. So I think it's a very interesting an allegory between knitting and the complementation pathway and genetics. I think that's very interesting here. And if only I could share screens, but I haven't figured out how to do that. But Robert here, her friend, that's also doing the demonstration. He looks like he's like a master of knitting. Let me just say that.
But by himself, he has no yarn, so he
Oh
can't
man,
knit.
poor Robert.
So alone, neither of us are able to knit. However, if we team up and we work together and we share our resources, now we have both yarn and needles, so we can knit a nice scarf. All right, so how about the case of non-complementation? If I have needles now, and Robert also has needles, me by myself, I cannot knit because I have a mutation in that I have no yarn. Robert also has needles, and he also does not have yarn, so by himself he can't knit either.
Alright, that makes sense.
This time, when we team up and we try to work together, even if we share our needles, we still can't knit because we have no yarn.
Alright.
So this is the case of non-complementation. Thanks, Robert. This concludes the help session on complementation. Thanks for joining us.
Alright, that made good sense where they related it to genetics and the pathways used to synthesize something. Alright, let's go to the second thing. Looks like this one I'm supposed to be reading it, so who knows what we'll find here. I know this looks like a Zoom class basically, but I just wanted to make a just chatting stream while I'm doing homework, so if anyone wants to ask questions or just chat in the chat box, I'm very happy to respond to any of those, so please do. Uh-oh, the server cannot be found on the second item of the list, so I guess we're just going to go to the third things. So what happens when I click that? It says Safari cannot open the page. So we're gonna have to do the third thing, I guess, instead of the second thing. The second thing actually looks interesting, but it doesn't like Safari for some reason. So let's go to number 3 on the checklist here of our journey into genetic complementation for this homework assignment, where it's saying read the textbook, half of a page on complementation in the textbook. I actually probably should have brought my notes for this, now thinking about it. And no materials are available, so I'm going to have to look for this. I don't have the textbook, unfortunately. Let me go in to try to find it. This is a little bit of a scavenger hunt, I guess. A little bit of a side quest, but it looks like I don't have the textbook either. There's nothing in here that's talking about the textbook. Safety form. Yeah, I don't think there's anything there that has the textbook in it. Unless that's the same chapter that has ours. We could kind of do this already. I think the teacher even went over it in class, but I could walk through it again. Let me just see. Is this a text box? Let me see. Nope. It has to be a... It can't be a text box. Alright. So let's just... I guess I'll just open, like, Ibis Paint or something like that. I'm good with drawing in Ibis Paint. Let me find my stylus. I have a nice golden stylus here for my phone as well as any of my tablets and my laptop that has a touchscreen. There it is. Okay, now let's go to the App Store, and I guess I need to download Ibis Paint, so let's do that. We're just going to be using this for like notes and whatnot because I don't feel like getting Adobe, so we're not going to do that. Of course, I forget my password. You know what? Let's not do this right now. I'll just go and use Adobe or take pictures of it. Maybe that might be easier. I could upload them tomorrow. Let's do that. Forgot my Apple ID password, so I'm not gonna even trial on that. Okay. I'll just read out what we're doing in class. It might be interesting to some of you. Okay, so again, this is about genetics, and we're crossing mutants, which are organisms that have the recessive trait for something versus the wild type, and we have different mutants, and we're trying to find out which one has a functional gene and where. So basically the gene will code for this enzyme in the pathway that we're trying to get to starch. It looks like in the problem we have sucrose, a bunch of different steps that require enzymes, and then the end goal is starch. So we're going to be looking at some organisms that use sucrose to turn into starch. Basically, it looks like we're going to be looking at a type of corn, so different specimens of corn that we're going to be testing for different alleles that may code for a functional gene for the specific enzymes, or if they're recessive, they're not going to be. And I guess some important words to know are mutant, which is, again, an organism whose allele has the recessive form of it, so recessive trait. Wild type will, of course, be the one that expresses the dominant phenotype. It doesn't have to be homozygous dominant. It could also be heterozygous dominant, or heterozygous, so it just needs to have that dominant phenotype being expressed. So I guess I'll just start here. We have sucrose, let's just say enzyme 1. Then we have something called UTP glucose. And then we have enzyme 2. Then we have ADP glucose. Then we have enzyme 3. And then we get to our end, which is starch. So if a mutation occurs in any enzyme, then sucrose instead of starch is deposited in the kernel. So again, we're talking about different species of corn that we're testing in this hypothetical problem. And the kernel is wrinkled. So if the kernel is wrinkled instead of plump, this means that one of the genes isn't functioning correctly, or it has the non-functioning gene due to it being a mutant. So we could either have a non-functional gene for enzyme 1, enzyme 2, or enzyme 3. And our job is to go to these scenarios where we're crossing like two homozygous mutants, or a homozygous and a normal kernel, which will get starch. So if it's a normal kernel of corn on the plant that we're looking at, that means starch is being produced correctly from the sucrose. It has all those enzymes working. And in turn, it means we only have the dominant alleles present. Or we could also have heterozygous. But usually, I think in this one, it means it's going to be homozygous dominant for that normal kernel. Okay, so I'm just going to write out again. I'm going to like scan what I'm drawing on the side for tomorrow when I actually turn it in. So let me just grab a quick tissue because my nose is running. Guess I'll just turn off the fan. I think that makes more sense, you know. I'm going to turn off the fan before I start sneezing. I don't want to sneeze on stream. My sneezes don't sound cute or anything. They sound horrible, so I don't want to sneeze on stream if I don't need to. Alright, let's title this like that. My laptop has shut down on us, but got it back working again, so that's good. Alright, so we have sucrose. And then I'm just going to do like SS. SS is like the name, the simplified name of the first enzyme. And of course enzymes end in an ASE, so it's SS-ACE. That's just again a simplified version of the name of it. Then UTP glucose. Enzyme number 2 is PPASE, PPASE. Then we go to ADP glucose. Then we go to GBSS-ACE. These again are all different enzymes, so that's enzyme 3. And then after the ADP glucose is worked on by that GBSS ACE, we get starch, so that's our end goal. And then some important things to notice. Capital S means a functional gene that will code for that first enzyme that's needed in the pathway. So let's just make a note of all these alleles. And then the next one will be a capital P. That means a functioning P-pace gene. So, and then last but not least, we have that GBSS ACE. So capital G, that allele will stand for functional GBSS ACE gene. Okay, so scenario 1. I would say, actually, we would actually have to put all mutations are fully recessive, so mutations are all recessive. So that just means the lowercase allele, so non-functional genes for those enzymes, respectively. Okay, we also need to make note when a kernel is wrinkled. So actually, wrinkled to kernel equals no starch. So that means one of the genes aren't there for one of the enzymes because we can't get to our starch product if all of the pieces aren't there. So need to make note of that. No starch. Alright, scenario A says if you cross two homozygous SS ACE mutants, again mutants means it's recessive, will the kernels be plump or wrinkled? Again, plump means starch containing, so we successfully went through that pathway, no problems at all, we have all the enzymes, and we got to starch at the end. Or wrinkled, where one of those are missing, and we don't get to our starch. So show the cross and the outcome, explain why. Alright, so two homozygous SS ACE, that will be 2 lowercase s times, actually, yeah, 2 lowercase s crossed towards each other, so then kernels will be wrinkled. So wrinkled kernels will occur since the outcome or the cross only results in plants or corn plants, but I guess offspring or individuals. That would display the phenotype of a non-functional SSACE gene. Lack of one of the enzymes needed in the pathway for starch. And then, of course, the Punnett square will be like a 2 by 2. I'll have S, S, all lowercase s's, and then, of course, will just be everything is SS. Alright, So that was scenario A. Let's go to scenario B. We're almost done, which is good because I need to... Sorry, excuse me. I need to go to sleep soon because I have a class early in the morning. I'm gonna actually turn back on the fan even though I'm getting a runny nose. Because it's getting hot in here. Alright, sorry about that interruption. Alright, let's look at the second situation. If you cross a homozygous SSase mutant with a homozygous GBSSase mutant, will the kernels be plumped or wrinkled? Alright, so this is kind of like in the scenario about knitting where one person didn't have needles, the other person didn't have string. So I'm guessing that this will also result in kernels that are wrinkled because you don't have a functioning SSase gene, nor do you have a functioning a functioning GBSSase gene, because those are both mutants that you're crossing, so I don't think either of them will be in play. So we have either are going to miss one of the enzymes, enzyme 1, or we're going to be missing enzyme 3 in the pathway, and that's not going to get our starch. And the kernels will be wrinkled as a result, so... I'm just going to say wrinkled kernels will occur because the offspring will be... There isn't a presence of a functional gene that's being coded by a dominant allele or something along those lines. That's what is going to happen in the long run anyway, so... will lack an allele codes for a functional gene. Needed pathway for starch. Therefore, the kernels will lack the ability to produce starch. Okay, so now I just need to shift across. So I might need a Punnett square, but we could do this. It would be SS times lowercase gg, and either where you go, you are going to get situations where it's just going to be homozygous recessive. And those recessive genes, because they're mutants, they're going to have recessive genes. We know the parents are, and you can't get the functioning gene, which is on the dominant allele, if you only have recessive ones in the cross. So this is going to result into SSGG everywhere. Okay, I actually forgot to get something. I'll be right back. Okay, I am back. Sorry about that little interruption. Okay, so the cross will be SSA, so it'll be like this. SSGG. Actually, got that wrong. 2 S's on the top, 2 G's on the side, the Punnett square. Just do it like that. Alright. It'll be wrinkled. I might want to write with a pen next time so it actually shows up. I just thought about this. It might not show up when I take the picture or scan the piece of paper I'm working on because it's also lined, and the table I'm writing on, it doesn't do well with these Ticonderogas, which are softer leads. The pencil isn't that dark, but I'll see what happens tomorrow. We'll just have to see. Alright, this scenario C, if you cross a homozygous double, one that has a non-functional SS ace and a non-functional GBSS ace with a homozygous GBSS ace mutant, will the resulting kernels be plump or wrinkled? So, homozygous double mutant, so non-functional SS ace, so that means homozygous both lowercase s in its genotype. Then we go to a non-functional GBSS ace in the same organism, so we just add the 2 G's at the end, and then we multiply it by GG, 2 lowercase g's, so everything is recessive. We won't get a pathway to starch, so these kernels are going to be wrinkled. Due to the resulting types lacking the alleles to code for one or more, I should say one or more, because one of them actually has 2 non-functioning genes. Functioning of required enzymes for the production of starch. Okay, so that was scenario C. Now we go to scenario D. This one's actually interesting. I remember it from class. If you cross a homozygous triple mutant, so again, a mutant is something that's all recessive, so excuse me, sorry about that. It would be lowercase s, s, lowercase p, p, and lowercase g, g. So that is our homozygous triple mutant times our homozygous normal kernel. So the homozygous normal means homozygous dominant. And it's not a mutant, so this is going to be our wild type. It'll have the phenotype of having the functioning genes for all 3 of those required enzymes. So this one I think will be plump, because the resulting kernels are going to be heterozygous, and that heterozygous does contain that dominant allele, which will result in that functional gene for all those enzymes, and we'll get to our starch. So let's write that down. So homozygous normal, again, all capital letters, aka dominant alleles. So since the offspring will be synthesis. Their genotype will have the presence of the dominant alleles that code for the genes for the enzymes needed in starch synthesis, which will allow for the offspring or the kernels to produce starch. Alright, so that was the assignment. We finished it. So I just need to snap a picture of that for tomorrow when I go to class. Alright, let's see what else I could do while I'm up here. You go to sleep at 10:30, so maybe like 15 minutes more of something. I'll try to do more of these streams where I'm just doing homework live on Twitch, I guess. These might be interesting as the year goes on. We might cover some very interesting topics. So yeah, I'll try to do more of these. So I'll change the locations around my room too, so the background isn't just my shelf of gemstones and Pokemon cards and stuff. So I'll do that. I'll try and continue the series as the year goes on. Let's go and let me see if the textbook for this other class is actually working or not, and we'll try to do some notes for it. We won't get to it all, but we might be able to do a little bit of notes. Alright, now I just need to go to the syllabus and see what chapters are needed. Of course, it doesn't want me to view the syllabus. There we go. And I just need the schedule. Come on, where is it? Here we are. It looks like we need to read chapter 1. So let's go and start on chapter 1. I already am taking like lecture notes online, but I also like to take a little bit of just quick notes of things that aren't review from previous biology and chemistry classes on paper, so I also have that as a copy to look at. Preparation for the test and whatnot in the class. Okay, so chapter 1, that looks like it's all this week. It's just introduction stuff, so it might all be a review, but I'll just glance through the textbook. I only now write things down that are completely brand new for taking notes. I think it's much more helpful and more concise that way, so that's what I've been doing. Yeah, just intro to biochem, so that's the first. It seems to be just the organelles and different things, nucleotides, maybe the macromolecules, and yeah, let's start doing this. Let's start on these notes. Let me see how I could actually get to open. Alright. So I got the I can't even open the book, it looks like. Saying a publisher access code is there. Interesting. Okay, let's try it this way. Let's go the back way here. Nope, still not letting me get in. So I guess we're gonna cut it here, I guess. I can't even get into the textbook, it looks like, unless she linked it somewhere, unless the professor linked it somewhere else. Let me try and find it before I give up. There's the Eve book. I don't think that's it. Checklist. Chapter 1. I think this is her slide. It's not actually from the book. Yeah, that's her slideshow. I'm looking for the actual textbook link, and I can't find it. Resources. Will this take me to chapter 1? Oh, no. That's just Khan Academy. We don't need Khan Academy yet. That's just the functional groups, and we know those off the top of our head because of organic chemistry, so I don't think I need that right now. I do just need the textbook link. Try and get it. Should be in course materials. I guess I can't access it yet. It's not letting me even click on it, so maybe right now we'll just end it here. We got something done at least, which I'm very proud of that we did that. We need to start actually reviewing amino acids, so that will be some of the streams, I guess, of this series of doing homework on Twitch. For one of the classes I'm taking biochemistry, so major component of that is memorizing all the amino acids, so on these streams I'll start doing that. I think I remember some of them a little bit. Some more than others, but we didn't really do a memorization thing in organic chem over them, but I don't know. We'll just have to see what happens. Adaptive quiz. Those aren't due yet, so I'm not going to worry about those. It looks like it's just a lecture for right now. I think everything else is done. I think I finished all my homework, it looks like, until we'll have to see tomorrow if I missed anything. Hopefully not, but I think I got everything. I think I did that. Okay, let me check. I'm just checking through all my classes, just in case I missed something. Pre-lab quiz, August 30th. Due August 30th at 8 a.m. That's a long time till then, so I think I could just wait on that. Let's go to assignments on this side. There's nothing there either on this. I don't see anything there. Let me see quizzes. I guess not. Interesting. Let's see the simple syllabus. These are supposed to be simple, so let's see if it'll have the information we need. Exam 1. Okay. Let's see. There should be like a... There we go. No, it's just... organization, human body. We did that today. I could start making a Quizlet for that. Probably that'll be a good idea. On Thursday we go over cells. There's just a test on Thursday. Then I remember on the labs, those practicals are actually going to be on the campus but over the weekend, so I need to watch for those. So I think we're good there. I checked genetics and I was just in genetics, so I think we're all good on that. Let me see assignments again, just in case. August 24, that's tomorrow. Section 1, that's just, I'm guessing that's like chapter 5, which I did read through. It was mostly review. Okay. Required lab, we were just there. Biochemistry, let's see, biochemistry lab. When did I even have this class yet? I can't remember. Guess not. Oh wait, pipetting. When do I have this class? I don't think I've even been to Chem 320L. Let me see. I'm just trying to check something. I don't even remember going to this class. It's 2 days and I still don't even remember. Let me see. Wednesday. That's why. Okay. It's Wednesday. So tomorrow. That's why there's nothing there. Okay, so that makes sense. Well, it looks like I got everything I need to done. And I need to go to sleep soon anyway, so I think this is where I'm going to stop the stream. Thank you so much, everyone, for viewing either live or later on. I really appreciate it. I hope everyone's doing well with the new start of college and or school. And other than that, I think this is a good stopping point. So thank you so much, everyone, for tuning in. Please do something nice for someone and get vaccinated. And again, do a second thing for someone nice. Do a second nice thing for someone else, maybe someone different. Just keep doing nice things for each other. Yeah, so thanks so much again for viewing, and I'll see you guys all again soon. Thank you so much, and good night to all of you. Love you guys. See ya.